Hello and welcome to the July 2023 Longevity Review, presented to you by the Canadian Longevity Association, a federally incorporated nonprofit whose mission is to help accelerate the introduction of effective longevity treatments and to ensure their free availability to all Canadians under the healthcare system. My name is Chris Linnell, and I am the founder and president of the association. We are a volunteer-run organization and are always looking for more help. So if you would like to assist us in achieving our goal, then please get in touch via the links below this video. In addition, you will also find a link to the newsletter being discussed here. This month, we will look at the first use of the longevity factor klotho in old primates, progress in developing chemically induced partial epigenetic reprogramming, the first demonstration of lifespan extension in mice with heterochronic parabiosis, as well as another paper showing how extracellular vesicles mediate its effects, as well as also extend lifespan on their own. Don't worry, I'll explain all of these terms in the main section. And finally, how also being happy could help with your longevity. But first, some major CLA news. So after we finished our post-incorporation legal requirements and were thus able to open up a bank account, we have now set up a donation service via our new donation page on our website. If you take a look here, so just a little information, and if you click on the donate button, it'll take you to our donations page using a service called Zeffy. And the good thing about Zeffy is that they give 100% of your donations to us. So unlike other services like PayPal, which might take I don't know, 1 or 2% cut of the money you give, Zeffy gives 100%. At the end, they sort of ask for a separate donation if you feel like giving them a little bit of extra money, but you don't have to. And you can give one-time donations, monthly donations, or even a yearly donation to us, either the amounts that are set there or any amount you choose yourself. If you click on this link here, this takes you directly to that Zeffy donations form or page. Now, one thing is at the moment, our expenses are relatively low, primarily consisting of keeping and maintaining our website as well as a few other services such as a pro Zoom account. But as we grow in the future, we're going to basically require more funds. Firstly, to get a few more services to help with our social media outreach, and also just to sort of build up a fund for future activities. I mean, we have ambitions next year to host a conference and things like that. So as we are volunteer run, our finances are negligible. So any amount of money you can donate, it will be greatly appreciated and will be able to be put to immediate use. Anyway, enough of that plug. Let's get stuck in with the roundup. So the first paper we are going to look at involves the longevity factor klotho. So klotho was a gene which was discovered in 1997, I believe, and when it gets upregulated in sort of transgenic mice, their lifespan increases, and it also helps with sort of metabolism, insulin secretion, I believe, and also seems to keep the kidneys healthy. And in fact, there's been some studies show that humans which naturally have a higher level of klotho live long or healthier in certain aspects. Now, I should just mention that klotho comes from a Greek word, a Greek goddess, who sort of spun the web of life, so regulated how long we live. Anyway, so although it's been 25 years since that gene was discovered and seems to show some good effects in mice, there haven't been any human studies or any studies in primates. Well, this is the first study which seems to wants to sort of bridge that gap in our knowledge, and they basically injected klotho into older monkeys, older rhesus monkeys, who were sort of roughly around 21 years old, so the equivalent of a 65-year-old human. And let's go take a look at the paper. Yeah, they injected three different doses of Klotho into a small number of monkeys. I think there were, I'm not sure actually here, but not too many monkeys, but they injected either 5, 20, or 30 micrograms of Klotho, and wanted to see whether or not it improved the cognition of the monkeys. Now, firstly, let's take a look at some results here. So this, let's make this bigger. Actually, let's make it bigger, even bigger. Here it is. So firstly, see on the right here, just shows the different quantities of Klotho injected into the monkeys and the relative increase in the serum levels of Klotho in the blood. And so you can see there's 2 UG, 10 UG, 20, I'm not sure if that's actually U, but basically that means a microgram. So there are 1,000 micrograms in a milligram, if you really want to know what that is. Anyway, so they gave 4 different doses here. And you can see that the higher the dose, up to 20 micrograms, the higher the concentration of Klotho in the blood serum. And figure 2. Here's figure 2. So they injected them basically once a month, and then they tested their cognition, because Klotho seems to improve cognition in mice, despite not crossing the blood-brain barrier. So it has somehow, otherwise in mice at least, improves cognition. And let's take a look at this up next chart here. So again, showing the protocol, what they used, and I say 4 hours after injection, they tested the monkeys in terms of sort of a normal memory usage or high memory, sort of normal memory levels or high memory levels. I guess they had to find food under boxes or something like that, and they had to remember where the food was hidden, either under 4 squares, I guess, or 6, as are shown here. Anyway, you can see, well, a couple things. Firstly, so this is showing the 10 microgram dosage, and sort of improved compared to baseline and compared to vehicle, which means the control. High memory usage or high memory levels showed an improvement. Also for normal memory performance also showed an improvement, and this seems to persist over time. So they showed after 4 hours it improved, and this persisted over 14 or 23 days later. Now, the relative improvement, this is sort of interesting, is that like a lot of things in biology, more is not necessarily better. So if you look at G and H here, high memory levels on the left, normal memory levels on the right. This is the improvement in performance, and you can see that the 10 microgram level produced a significant higher level of improvement compared to the 20 micrograms or 30 micrograms. Indeed, under the sort of the normal memory load, once again, the 10 micrograms showed the best results. And in fact, 30 micrograms here seem to imply a negative effect on memory. So as I say, more quite often is not necessarily better. But regardless, is an interesting result. And I mean, the significance here is simply the fact that they are testing this in primates. So obviously, rhesus monkeys are much closer to humans than mice are. And the question has always been, if something works in mice, does it work in humans? The fact that this appears to work in rhesus monkeys is certainly just one step short of trying it out in humans. And as the researchers themselves say, the next step is trying this out in humans. But interesting, finally, I mean, it's always exciting to see any studies done in monkeys. And so that's why I wanted to highlight this research. Hopefully in a few years, we will see some results of human studies. Moving on to the next paper. So this involves partial epigenetic reprogramming, which I claimed earlier I was going to explain. I'll do my best for that. So basically, this involves methylation on top of our genome. So obviously, you know that our genome contains the instructions for all the proteins made in our bodies, but how does each cell has the same genome, but each cell produces different proteins. How does a cell know, like a heart cell or a brain cell, know which proteins to turn on? Well, it's these little what are called methyl markers or on top of the genome, which basically tells the cell whether or not a gene is turned on or off. And those markers change over time. They change as we age. And using the Yamanaka pluripotent factors, which basically, well, the way we create turn a normal cell into a stem cell is by using these Yamanaka factors, which basically sort of wipe clean or reset all those methylation markers and restore them to a youthful level. So we can actually take a cell from a supercentenarian of 110 years old, use these factors and basically revert the cell back to a youthful level, so that he couldn't really distinguish that old cell compared to a cell from someone who's 20. Now, the processes so far have used genetic techniques or transcription factors, which might not be able to be used in us, or it could be quite difficult to employ them in humans. So there's now a hunt on to try to find sort of chemical factors, sort of drugs basically, which can do this process without needing to worry about genetic manipulation, which carries the risk of causing tumors to form, which of course we do not want. Anyway, in this next paper, researchers led by David Sinclair and Vadim Gladyshev, I got his name wrong, from Harvard, have been looking into producing, as I say, a chemical means to reprogram or partially reprogram the cells. And this is exactly what they have done or made progress on, at least. Now, here's a word salad, or using a nucleocytoplasmic compartmentalization assay, which in a nutshell basically looks like as cells get older, there's a breakdown between the nucleus and the surrounding cell in terms of the proteins transferring between the two. And as cells get older, as we get older, that barrier, I guess, keeping the proteins on the right side sort of breaks down and I guess causes problems. It is very evident in senescent cells. Anyway, they use this word salad to screen various cocktails of drugs, or cocktails of molecules which have already been shown to have an effect on methylization. Now, they tested 80 different cocktails of these various molecules, and they selected 6 of those for further study. And let's go down to where's figure 4? Figure 4. just figure 4, figure 3. Let's keep on going down. Figure 4. Here we go. So they then tested 6 different cocktails of these various molecules on cells. This was all the experiments here were done in sort of cell culture using senescent cells. And if you look at these graphs here, you can see on the left, taking A as an example. So on the far left in the red bar here, quiescence. So that's sort of a kind of a normal cell. Oh yeah, this is biological age, or change in biological age on the left-hand axis. And senescent cells showed an increased biological age. Now, if you look on the right here, you can see the 6 different cocktails. They all reduced the epigenetic age of those cells by varying degrees, and they used here are 3 different clocks that were used, and they all showed a reduction in epigenetic age. And not only that, if you look at this sort of heat map here, and just briefly here, all these various different pathways and genes and stuff. At the bottom are in red are all the different sort of aging tissue, different parts of the body, and then on the right, in blue, those are using the Yamanaka factors. In black here are the 6 different cocktails, and you can sort of see there's a quite considerable difference in gene expression between old cells and these potentially rejuvenated cells using this chemical reprogramming. And basically, the changes seem to be that where is it gone? So things that that should be reduced got reduced. Things that should be improved got improved. Anyway, it's interesting to see how that well they seem to mirror to some degree use sort of the chemical reprogramming seem to mirror to some degree the genetic or Yamanaka factor reprogramming. And so you can see here just an F at the bottom, with reprogramming, respiration increased, inflammation went down, so the cell metabolism improved, inflammation markers went down, and implying there that there was age reversal. So what else? So yeah, so the transcriptomic age, according to this, these clocks and stuff sort of are the equivalent of a 3-year reduction in transcriptomic age. So again, this is sort of early days. They have the next steps involve actually testing these chemical cocktails in living animals, in mice. So this is a long way away from human use. But as the authors themselves say, that eventually this could result in a systemic treatment in us. I mean, we're talking at least 10, definitely more than 10 years, possibly maybe around 20 years from now. This will be available for us and could give us a systemic rejuvenation, which would be quite nice, in my opinion, at least. Continuing on now, using a completely different modality of rejuvenation, this one involving heterochronic parabiosis, another term I will try to explain. So heterochronic parabiosis involves basically joining 2 mice together surgically, or joining up their circulatory system, and the heterochronic aspect means mice of different ages. So say take an old mouse and a young mouse, join them together and see what effects it has. And the effects it has shown in the past are that the young mouse gets biologically older, whereas the old mouse gets biologically younger. Now, these experiments have been going on for quite some time. However, up to this point, nobody has attempted to see the effects on lifespan by using this procedure. And that is exactly what the authors here did. Now, I should just quickly say that this article is behind a paywall. However, I found the original preprint from a couple of years ago, which I should, as an aside, you see here, this was posted in November 2021. So you can see how long it actually takes from, or this is just an example of how long a new study or paper goes from being completed to being written to actually finally being published in a journal. So what you're reading in journals, or you're hearing about in these reviews, are sort of, a couple years out of date. So the science has probably progressed a fair bit further than what you're reading about today. Anyway, what they did in this study, so they co-joined roughly 20-month-old mice, which I think is like the equivalent of someone in their 60s. And they joined them together for joined them with young mice for either 5 weeks or 3 months. And the thing that they then did was, after 3 months, they surgically detached them, and to see whether the effects of being attached persisted, and also to be able to sort of have a proper lifespan study showing that just 3 months of this heterochronic parabiosis, this joining of the circulatory system, has a longer term effect. And they then follow them for 2 months later to see again if this effect persisted after 2 months of detachment. Now, what they found, let's get down to one of the figures. So here's a good summary of what they did. So here you can see that the procedure, mice are apart, then they get attached, circulatory system goes together, and they were detached 3 months later. Now, for the detached mice, in terms of lifespan extension, you can see here, the red arrow are the, sorry, I should just go back to the procedure. They co-joined. There are 3 groups of mice. One where they just had young mice attached to other young mice, old mice attached to other old mice, and old mice attached to young mice. So yeah, 3-month-old mice they used, and 20-month-old mice. So this red line shows the lifespan of the old and old attached mice. Now, obviously, this procedure is quite stressful on mice and stuff, so that's the best thing we can compare it to. Whereas this, whatever color that is, the other line shows the old and young mice. So the old mice who are attached to young mice, and they lived longer. So that led to a 6-week improvement in median lifespan, so average lifespan, and a 2-week improvement in maximum lifespan. So not a huge amount, but obviously a week for a mouse is a lot longer than a week for a human. And it also get rid of that. So, I mean, it led also to other improvements. So I should reduce their methylation age. So going back to epigenetic reprogramming, reduced their methylation biological age by 24%. And so in terms of the genes used, it upregulated sort of genes to do with cellular energy metabolism, downregulated inflammation, similar to the epigenetic reprogramming, and increased the TERT gene levels, which affect the telomeres. So longer telomeres might be better than shorter ones. So this is there. oops, it says there's reduced physiological aging, DNM, that's the methylation, DNM methylation age, and also transcriptomic age. Oops, get rid of you. Let's see, was there another one? Oh, yes. A couple more studies looking more specifically. Click on this. There we go. Make this bigger. Oops. Make this bigger. Okay, so what this is just showing is looking at various different organs. Actually, this is looking at the blood. You can see the vial here. And again, what this is showing is at least using sort of different clocks. It sort of shows that there was a reduction in this methylation age according to these various clocks of 19%, 28%, 27%, 19% again, which is obviously good. And here looking specifically at the liver. So here once, get rid of you. That's always going to come up. You can see once again there is, if you're looking at the bottom axes. So old ISO means two old mice together. Old het means young mouse and an old mouse. And if you look at this this graph, it shows that the old mice attached to young mice had a much greater lowering of their age according to this metric. And finally, because I mentioned at the beginning that they carried out this experiment in sort of two different time lengths, one where the mice were joined together for only 5 weeks, another one where the mice were joined together for 3 months. And what they found was, if you look at the left and the right, that there was a much greater effect with longer term treatment. So you can see here, 17.9% reduction and the methylation age. With long-term treatment, only 6.6% reduction for 5-week treatment. So longer is better. Now, you might ask, how is this useful to us? Since you're not exactly going to be co-joined with a young person. Yeah, I don't think you'd want to be attached to some young kid, drag them around. So why is this potentially useful for us? Well, the whole point is that if we can discover what the differences in the actual blood are between old and younger people, then we can maybe use other means of using that knowledge to not use young people, but sort of, I guess, manufacture the factors or the things which make us the older mice younger, and use that in ourselves. And in fact, that leads me on to the next study, which is here. So the final term I'm going to try to explain. So this study used young extracellular vesicles. So what's an extracellular vesicles? Basically, it's a means that our cells and our bodies use to communicate with each other. So one method of communication involves sort of releasing these sort of packets of chemicals, which are basically sort of packets of information, which cells release, and they circulate around our bodies via our bloodstream, and they influence other cells in our bodies. And actually, one of the detrimental aspects of senescent cells, so cells which have sort of gone rogue, turned into zombies, and are causing all sorts of problems, those problems, such as increased inflammation, are partially caused by these extracellular vesicles, also known sometimes as exosomes. And however, like these extracellular vesicles can either produce detrimental results or beneficial results. And when researchers gave young extracellular vesicles, which came from the blood of young mice. In fact, if we go down to the first figure, you can see here, they used the blood from neonatal, so I guess newly born mice, a very, very young mice. Sorry, not mice, rats. This study was done in rats. And they gave them to 22-month-old rats, and they gave them monthly injections over the course of, they gave them 4 monthly injections. And what they found, because, sorry, I should taking another sort of quick step back. So, extracellular vesicles aren't the only thing found in blood, but it is one of the primary constituents which affect us. And in fact, the researchers sort of did sort of 4 different experiments, giving just the young whole blood to the older rats, just gave them the serum from the blood to the older rats, just gave them the extracellular vesicles, which is what we're looking at here, and also gave them the blood without the extracellular vesicles. And let's take a look at the effects. Let's look at here. So the effects they had, firstly, you might not be aware, but our hearts tend to sort of enlarge as we get older. That sort of they get thicker and less efficient in pumping. And here, first result is that the blue is EV, so those are the extracellular vehicles. The red is placebo. And the mice, which, God, how do you get rid of this ad? Anyway, I can't get rid of this ad. That man is annoying. That's how we get rid of the ad. Okay, good. So the blue line is placebo, red line, sorry, other way around. Blue line is the EVs. We'll just keep on calling them that. So basically what this is showing is that the heart did not thicken as much with the extracellular vesicles. In fact, it got less thick. You can sort of hear a cross section. There's more gap with the EVs, yeah, the cross-sectional area was less with the EVs, and also the amount of fibrosis. So fibrosis is not good, and the amount of fibrosis was quite a bit less in the extracellular vehicle rats. And also here, this is showing the stiffness of the heart. So again, as we age, our hearts get stiffer, which is, again, not a good thing. And anyway, the divergence between the red and the blue lines indicate that the stiffness is less with the extracellular vesicles. Again, here, 2 more things showing it, 2 more measurements showing a difference and an improvement with the extracellular vesicles. And here, most less abstractly, figure 4A shows the exercise endurance. And basically, upon receiving the vesicles in the first month, their exercise performance improved. Certainly compared to controls, exercise performance was considerably better. And here shows the lifespan effect. So after 65 days, 91% or 92% of the rats given the EVs were still alive, only 57% of the placebo rats. So latency to death, how many was 54 days more for the rats given the extracellular vesicles. So almost a 3 times improvement there. And figure 4, and what else improved? So yes, so the treadmill capacity here, that's an improvement of 16%, supposedly. One thing to also again, sort of fibrosis is improved down here. One sort of interesting result was that actually when they injected the blood without the extracellular vesicles, actually senescent cell count increased, which was sort of an interesting observation. But let's just go down to this little nice summary. Here we go. So the structural and functional effects, fibrosis went down, exercise tolerance went up. Once again, in the heart, fibrosis, hypertrophy, so enlargement, stiffness, I'm not sure what BNP is, all decreased, and efficiency improved in the lungs. And once again, fibrosis decreased, exercise tolerance increased, and basically renal function improved. So yeah, we want less glomerulosclerosis, and glucose tolerance went up and lifespan increased. So this is some quite intriguing results. Now, another significance of this, like who cares? Well, about extracellular vesicles is that theoretically, and other papers have shown this, is that they could theoretically be manufactured en masse, like using a bioreactor. They could just produce copious amounts of these ones, sort of young extracellular vesicles, and so they could sort of be put on the shelf and injected into us when needed. So I don't know how far away. I don't know how far away this is to being tried out in humans, but still, I think it shouldn't be too far. I mean, I would certainly hope within the next 10 years, this is they are using these they're being tested in humans. So intriguing and certainly encouraging results. Now, the final paper we are going to looking at it briefly because there are no fancy charts or graphs to take a look at, is the correlation between happiness and lifespan. Now, in this study, which was done in Singapore, involved 6,000 Singaporean adults of various ethnicities, all over the age of 55, and they basically conducted a survey and asked them, how happy are you? Are you very happy, fairly happy, sort of neutral, fairly sad, or, very sad? And then they followed these people for 11 or so years and, saw what their mortality was. And so, 11.7 years of follow-up. And what they found was basically, can't find the can't find the data here. Basically, after 11 years, almost half of the very sad people had died. 2/5, so 40% of the fairly sad people had died. Only a quarter, 25% of the neutral people had died. 1 in 5, 20% of the fairly happy people had died, whereas only 1/6 of the very happy people had died. So levels of happiness seem to correlate quite closely with mortality. Now, this is a correlation study. It's not a causation study. It's not saying that, yes, if you're happy, therefore you're going to live longer. Maybe it's the other way around. Well, what they also seem to show is that happy people tend to be healthier people. So it's the fact that happiness and health seems to go together hand in hand. Therefore, obviously, if you're healthy, you're going to live longer. And let's see if I can find. Let's go down to the discussion. Yeah, implications of study finding. I'll just read out. I'll read this out for you. Simply put, our results suggest that happy people live longer because happy people are healthy people. Policymaking should realistically adopt tangible strategies such as preventing or alleviating, so reducing depression, improving physical and mental health, I did not mean to do that. Yeah. No, stop. Here we go. And psychological functioning and well-being. This promotes both healthy and happy longevity. So, I mean, you might say, well, this is pretty self-evident. How's this going to help me? I guess I just wanted to point out that we should strive for happiness. I mean, if you're having mental health issues in terms of depression, loneliness, I mean, again, loneliness has been shown to be as deadly as smoking. And lonely people aren't very happy. I guess the takeaway from this is, don't neglect your mental well-being. Don't neglect if you're feeling down. Reach out to somebody. Be social. Do things that you enjoy. It's summer now. It's actually a beautiful day out there. As soon as I finish this video, I intend to get out there and enjoy the sun with sun cream. Very, 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 very important. So yeah, it was, as the song goes, don't worry, be happy. And not only will you be happier, but you'll also hopefully live longer as well. Okay, that was the final study we're going to look at in depth. I just wanted to point out that there were a number of very interesting exercise studies, which you can find in the exercise section of the supplemental section. And without going into all of these, you can take your time yourself. I will just give you a few high-level things to the too long to not read TLDR. So one interesting study showed that simply a single 3-second eccentric contraction 5 times a week can grow muscles, and it's important to grow muscles for longevity. So what does that mean? So looking at my arm, this motion, massive muscles here. This is a concentric contraction. Doing the other way, sort of loosening, or this is an eccentric motion. So simply if the 3 seconds, so 1, 2, 3, I guess with a weight, doing that 5 times a week will actually result in muscle growth. Secondly, another study looking at what types of exercise are better for different aspects of resistance training. So higher loads were better for increasing strength. Higher number of repetitions, the number of times you do the exercise, were better for increasing muscle size. Also, another study showed that only exercising on weekends is almost as good for your cardiovascular health as exercise spread throughout the week. So for those weekend warriors amongst you, you're doing yourself a lot of good. And finally, also, in terms of reducing blood pressure, which is incredibly important for our long-term health and longevity, all exercise is good. However, planks and wall squats are best at reducing blood pressure. They reduce blood pressure by the most amount. But however, the biggest take home from all of these studies is just simply, for the most part, any exercise is better than no exercise, and the more you do, for the most part, is better than the more you do, the better. And in fact, the study which was looking at the types of exercise points out that they don't want to sort of complicate matters for people. Just do what you can, and try to find something that you enjoy. Any exercise is better than none. And it's summer now, so get out there and do some exercise. Just go for a stroll, do whatever you enjoy, because exercise also improves happiness. That ties in well with our last study. Okay, that is all for this month. I guess I should just sort of say once again, please, if you like this video and you want to support the Canadian Longevity Association, then please take advantage of our new donate buttons. You also find link to our donations page below this video. Please like our video, like this video for the Google algorithms, and please subscribe to this channel so that you will not miss any further or future videos. So all that remains for me to do is to say goodbye and wish you all the best of health. Goodbye.